Chapter 4: Evolution and Diversity of Woody and Seed Plants. Lignophytes, woody plants. The lignophytes or woody plants, also called lignophyta, are monophyletic lineage of avascular plants that share the derived features of a vascular cambium, which give rise to wood, and a cork cambium, which produces cork. Figures 5.1, 5.2. These features also occurred in now extinct lineages with the lycophytes, example, Lepidodendron, and ecpistophytes, example, calamities, but are thought to have been derived independently in this taxa. A vascular cambium is a sheet or hollow cylinder of cells that develops within the stems and root as a continuous layer between the xylem and phloem in extant histidly. Spermatophytes. See later discussion. The cells of the vascular cambium divide mostly in the tangential plane, resulting initially in two layers of cells. Figure 5.3. Of these layers remain as the vascular cambium and continues to divide indefinitely. The other layer eventually differentiates into either secondary xylem wood if produced to the inside of the cambium, or secondary phloem if produced to the outside of the cambium. Figure 5.3, 5.4. Generally, much more secondary xylem is produced than secondary phloem. As secondary tissues is formed, the inner cylinder of wood expands. Figure 5.4, 5.5. Many woody plants have regular growth periods. Example, forming annual rings of wood. Figure 5.5. A cork cambium is similar to vascular cambium, only it differentiates near to the periphery of the stems or roots axis. The cork cambium and the derivatives constitute the periderm, referred to as the outer bark. The outermost layer of periderm is cork. Figure 5.4. Cork cells contain a waxy polymer called suberin, similar to chitin, that is quite resistant to water loss. See Chapter 10. The vascular cambium and cork cambium were a major evolutionary novelty. Secondary xylem or wood function in structural support, enabling the plant to grow tall and acquire massive system of lateral branches. Thus, the vascular cambium was a precursor of the formation of intricately branched shrubs or trees with tall overstory canopies. Example, figure 5.2. A significant ecological adaptation cork produced by the cork cambium function as a thick layer of cells that protects the delicate vascular cambium and secondary phloem from mechanical damage, predation, and desiccation. Woody can be quite complex. The details of cellular structure are important characters used in the classification and identification of woody plants. Woody anatomical features may also be used to study the past, a specialty known as dendrochronology. See Chapter 10. Spermatophytes, seed plants. The spermatophytes or seed plants, also called spermatophyta, are monopolytic lineage within the unlignophytes, figure 5.1. The major evolutionary novelty that unites this group is the seed. A seed is defined as an embryo, which is an immature diploid saprophyte developing from the zygote, surrounded by nutritive tissues and enveloped by a seed coat, figure 5.6. The embryo generally consists of a immature root called the radicle, a shoot apical meristem called a epicotyl, and one or more young seedlings, the cotyledons. The transition region between root and stem is called the hypocotyl, figure 5.6, 5.12. An immature seed prior to fertilization is known as an ovule. Seed evolution. The seed evolution evolved several steps. The exact sequence of these is not certain, and two or more steps in seed evolution may have occurred concomitantly. Figure 5.2 composite photograph of Sequoiadendron giganteum, a woody conifer that is the most massive organism on Earth among the tallest of trees. The probable steps in seed evolution are as follows. Number 1. Heterospory. Heterospory is the formation of two types of haploid spores within two types of sporangia. Large, fewer numbered megaspores, which develop via meiosis in the megasporum rangium, and small, more numerous microspores. The products of meiosis in the microsporangium, the ancestral condition in which a single spore type forms, is called homospory. Each megaspore develops into a female gametophyte that bears only archegonia. A microspore develops into a male gametophyte bearing only aphedria. Although heterospory was prerequisite to seed evolution, there are fossil plants that were heterosporous but had not evolved seeds. Among these being species of Archaeopteryx. Note that heterospory has evolved independently in other seed plants. Example, in the extant lycophytes Selaginella and Isoltes, and in water ferns. Chapter 4. Endospory. Endospory is the complete development of, in this case, the female gametophyte within the original spore wall. Figure 5.7. The ancestral condition in which the spores germinates and grow as an external gametophyte is called exospory. Reduction of megaspores number to 1. Reduction of megaspores number occurred in two ways. First, there evolved a reduction of the number of cells within the megasporangium that undergo meiosis, each termed a megaspore site or megaspore mother cell. Was reduced to 1 figure 5.7. After meiosis, the single diploid megaspore site give rise to 4 haploid megaspores. Second of the 4 haploid megaspores produced by meiosis, 3 consistently about, leaving only 1 functional megaspore. This single megaspores also undergoes a great increase in size correlated with the increased availability of space and resources in the megaspore rangium. Retention of the megaspore. Instead of the megaspore being released from the sporangium, the ancestral condition as occurs in all homosporous non-seed plants. In seed plants, it is retained within the megasporangium, figure 5.7. This is accompanied by a reduction in the thickness of the megaspores wall.